Hello everybody and welcome to this webinar which is called The Science of Baking, It's in Our DNA. We're here at the Science Kitchen at the University of Leicester. I'm Josh Smalley. You'll have seen me maybe on the Bake Off in 2023 getting through to the final, but I also work here at the university as a science communicator but also as a chemistry researcher in the chemistry building. I've been here now for around about 11 years. And what we're trying to do with these webinars now is give it a great opportunity to showcase these two worlds of bringing science and baking together. And today I'm joined by a very, very special guest for this webinar, and that is Dr. Syabira Yusoff, who was on the Bake Off in 2022, got all the way through to the final and won it. And it's an absolute pleasure to welcome her here today to deliver this amazing webinar. And as you can see, we have already got a creation made for you in front, which is inspired by your semi-final creation?
Yes, it is indeed by the semi-final creation. We've been tasked to actually build something as the most achievement or most memorabilia that we want to create it. And this is what I made, which is DNA. Very close to my research, very close to my heart.
Lovely that you were able to bring in your own sort of like career into it, onto the Bake Off, which is lovely to be able to see science shown on the television as well. So also today, it is International Day of Women and Girls in Science, so an extra special day to be able to have Syabira with us. And we're going to talk about this structure in a little bit more detail in a moment, and the theme of DNA is going to run all the way through the webinar. But before we do that, we've actually got a really amazing little message from our Chancellor here at the University of Leicester, which is Dame Dr. Maggie Addering-Pocock. So I've got a little video to show you now, and after we've done that, we'll go through the structure in a little bit more detail.
Hi, I'm Dame Dr. Maggie Addering-Pocock. I'm very proud to be the Chancellor of the University of Leicester, and I'm a space scientist and a science communicator. I think it is incredibly important to include girls and women in science and STEM, science, technology, engineering and maths. It's important because we need a diversity of different ideas coming together, and we need to see things from different viewpoints. If we have just one small demographic doing science, it doesn't work for humanity. Two of my heroes of STEM, science, technology, engineering and maths, are Gladys West. She looked at the shape of planet Earth, fed this into the GPS systems, which gave us much more accurate predictions of our locations. The second hero is Margaret Burbridge. She looked at the chemistry of our local star, the Sun, and gave us an understanding of why it shines so brightly. I hope that everybody really enjoys the webinar from the Science Kitchen.
How wonderful was that? That was really special to be able to have Maggie to be able to deliver the little introduction for our webinar. So back to the creation that we've got in front of us. I think Syabira wants to add a few more words around what was the inspiration behind making this on the show.
Yeah, so when I was on the Bake Off show, like I said, DNA is very close to my heart, and I would like to tribute to the Rosalind Franklin, that without her, she's a biochemist as well, actually, and she's based at the King's College London. And without her, this discovery of DNA might not even be there. So, yeah, what I tried to mention is with the DNA helix structure, you can see it's very impressive helix, but actually it got much more information towards it. Yes. Now rotating it, you can see the phosphate bonds around it, and then you can see different colors that we put together. So this is actually in the entire human genome, you only have these 4 letters, which is G, A, T, and C. So those 4 colors are representing to it. And I can't wait in our webinar later, Josh, we're going to discuss further how this GATC translated into making protein, and how even in the baking, everything is actually science.
It certainly is. And actually, when we were putting this together yesterday, it was so much fun because it felt like a bake-off day yesterday because we were putting this all together. And we actually did it in around about 3 hours, 4 hours. So we did pretty well. Actually, I've got one of the biscuits left. So we've got 3 of these biscuits like this that are a bit of a structural support, which are going through the middle. And then we've got all these, there were 14 centimeters length, very accurately measured out, as scientists is what we have to do. And then we've been putting them all together as we've been going up. As you can see, we've got the colour, different colours all going around it. We've got a nice blue on the outside. This is to showcase our sugar phosphate backbone, which is what we've got that makes up our DNA structure. And then, as Syabira's already said, we've got the colours going up the middle, and that's to showcase all of the nitrogenous bases that we've got that make it up. And these ones have particular ones that link together. So I think what we'll do now is I've got a few slides now to go through this structure in a little bit more detail so you can see it accurately, not just in bake form. And then whilst I do that, Syabira is going to get set up, because what we're going to do is we're then going to go into doing an actual baking demonstration. We're going to do some science of baking. We're going to make this lovely cake like this just here. And we're going to be talking about all the science that goes into behind creating a bake like this. And in particular, the flavour we're talking about today is the strawberry. And the strawberry, what we're going to do, we're going to actually do a DNA extraction demo as well as that, so we can see DNA live, and then we're going to also use the strawberry to mash it up, make a jam out of it for the cake. So I'm going to go through a few slides now. I shall leave Syabira to get set up.
Yes.
So if we go through to the slides now, the DNA structure in a little bit more detail. So here is the DNA double helix, as you can see on this left-hand side. And what I'm going to do now, we're just going to zoom in to a short section of it so we can look in more detail at the actual, on a molecular level, how this is made up. So as you can see, what we've got here is we've got down the left and right-hand side, you've got this sugar phosphate backbone. That's those pentagons and those circles that are linked together. And the DNA itself is made up of these nucleotides. So the nucleotides is in the top right of your screen there, and you'll see that each nucleotide, we've got a pentose sugar, we have got a phosphate group, and we've got a nitrogenous base, and they're all linked together to make up our DNA structure. And what we have is we have one long nucleotide sequence, we have two of those strands, and those two strands together join up and they make what is our structure of DNA. So if we go a little bit further now, we're going to talk about the nitrogenous bases. As we've already alluded to, you can see the colours that we've got right here on this slide are the same colours as what me and Syabira have put onto our bake. So you can see we've got adenine, thymine, guanine and cytosine, and they are those 4 nitrogenous bases that we have in our DNA. So it's only made up of those 4, and those have such a unique sequence, and that unique sequence codes makes us the unique DNA that we have to each of us, and they're going to code for the synthesis of all the proteins that we have in our body, and that's our unique genetic code to each of us, which is passed on from one generation to the next. So we're going into a little bit more detail how this structure is put together. So what we've got is you can see the little red dots that are going across from left to right. You'll see the little hydrogen bonds. I've indicated them on the bottom right of your screen. So those hydrogen bonds, they are formed between these base pairs. So between adenine and thymine, we've got 2 hydrogen bonds. And I'll go through a little bit more detail on the structure of those on the next slide. And then guanine and cytosine have got 3. So these are the hydrogen bonds that's helping to keep these 2 strands together. And then also indicated on this slide, you can see we've got some hydrophobic, some base stacking. This is stacking vertically now between those bases. But these are called van der Waals interactions, and they're also helping to keep this DNA structure together so that we have that DNA double helix. So if we now go and look at this, this is now in the chemistry form now. So some people, if you've got a chemistry background, interested in chemistry, you may have seen these before, although you may not have just seen the form that I showed you on the previous slide. But don't worry, I'm going to go through this in great detail, all colour coordinated, so you can see how this links up to that previous slide. So this is now on the chemistry level, all the atoms now and all the molecules that make up our DNA. And if I go through same, you can see on the top right, we've got our nucleotide again, still the same pentose sugar, but now just the actual molecular structure of it. If I now highlight these, what you can see, we've got this sugar phosphate bonds that are going up and down the left-hand side there. These are these 2 strands. They run anti-parallel to each other. So if you can see, we've got a 5 and a 3 running from top down to the bottom on the left, and it runs the other way on the right-hand side. And these are all linked by phosphodiester bonds. We've then got our different nitrogenous bases. So you can see we've got thymine indicated there in orangey yellow. We've got our adenine there. We've got our guanine and we've got our cytosine. And they're all linked up together, and you can see indicated the hydrogen bonds as well between each of those. So you can see where those hydrogen bonds are forming, and those hydrogen bonds are forming between a hydrogen and a electronegative atom. So in this case here, it's either oxygen or nitrogen. So you can see in each case that hydrogen bond is going between an H, a hydrogen, and an oxygen or a nitrogen. As I said, these are running anti-parallel. The reason why it's 5 to 3, so you may, if you've been interested in biology, from a biology background, you'll know about these 5 to 3 sequences. But you can see now from a chemical point of view why it's 5 and 3, because you can see the numbers that I've got now on that top right nucleotide structure. It's atom number or carbon number 3 and 5, and then you can see on the strand in the middle now where we've indicated 5 ending down to 3. So that's where those numbers come from. And then also, I can talk a little bit more detail about these purines and these pyrimidines, because these are what are our nitrogenous bases. They're in 2 different forms. So we've got the purines are these double rings. This is our adenine and our guanine here. So you can see we've got a 2-ring structure, and then the pyrimidines are the thymine and the cytosine, and there's the red and the orangey ones there. And you can see the pyrimidines, they're just a single ring. They've got a 1,3 nitrogen in, and the purines they look like a pyrimidine with another ring fused to it. So that's what we've got making up the structure of our DNA. And now a couple more slides just to finish off, so just to sort of relate it back to the bake that me and Syabira have made. So I mentioned that this DNA structure is stabilised by hydrogen bonds, van der Waals interactions. The most common form of DNA that we see is bDNA, which is the model deduced by James Watson and Francis Crick, often referred to as that Watson and Crick model. This is bDNA, so this anti-parallel, this right-handed turn. You can see it there shown on the axial view looking down. But also there's not just that form, there are other forms that are present as well. So here's some structures now showing you aDNA and zDNA. And these ones are formed depending on the sequence of these nitrogenous bases that we've got in our DNA. So you can see there, if you look on the bottom left, with aDNA you have stretches of the purines. So that's so for example, if it was GA, they're going to have this aDNA form. You can see it's slightly different to B. And then if it's a Z, you can also have a zDNA form, which is where you can have alternating pyrimidine and purine units. So for example, CG. And in that case there, we end up actually with a left-hand turn this time, and you can see that it takes the form of that zDNA on the right-hand side. So that's a little bit more detail on the structure of the DNA. Now we're going to go on to a baking demonstration. So what we're going to be doing is we're going to be making a strawberry and cream Genoese sponge. Here is the ingredients list. We're going to go through this in some detail now. We can probably keep referring back to this slide so we can sort of talk about each of those ingredients in person, and then we're going to go through to the demonstration. So hopefully I shall now head back over to see Syabira, who has now prepped us, and we're now ready to go for the baking demonstration. So lead it away. What have we got to do first?
What we're making today is, as Josh mentioned before, is a Victoria sponge cake, and we decided to go with the Genoise sponge cake,
Bake
which
off.
is yes. So this Genoise sponge cake is very airy, very light, and we're going to introduce how this airy and light, in terms of the chemistry, how it's created a very good it's a meringue.
Yeah, meringue, yeah.
To create a very airy structure of the cake. So what we have here is the eggs. We have separated them to make the white eggs for the meringue. And eggs is very important for the stability and the structure for the cake. And then we have flour here, interacting with the moisture to create the structure as well. And you want to mention something about the leavening agent?
Yes, so this is using self-raising flour. So as Syabira said, we're going to be making a meringue. So we want to get this lovely airy structure in our cake. So we're going to do that in 2 ways. We're going to have a meringue in here where we're going to whisk that up with this lovely light foam. So we've got air bubbles that we've incorporated in. And we've also got our self-raising flour that actually has got, self-raising's got a little bit of baking powder in it. I've actually got the packet here of the self-raising flour that we use. And you won't be able to probably zoom in, but I can read out what you've got in here. And it says that we've got raising agents, we've got sodium hydrogen carbonate, which is bicarbonate of soda. So that's what you'll have used in cakes, meringues, all sorts of things. And it also says we've got disodium diphosphate and we've got monocalcium phosphate. Now, if you've not seen these names written down before, they'll look quite foreign and you might not think much of them. But what they are, those are the two that I mentioned. They're both acids. And what happens is when we've got the acid mixed with our bicarbonate of soda, when they go into liquid form, they react together, they form carbon dioxide bubbles. Also, the bicarbonate of soda, when it goes into the heat of the oven, it also produces carbon dioxide bubbles. So we've got some aeration from the little bit of leavening agent in there, but we've also got our meringue as well.
But since we mentioned about the leavening agent as well, when we have the whisk meringue, this recipe works with self-raising flour. Even if you don't have self-raising flour, this gonna still works. We're gonna show you. Yeah,
but if you're gonna do it without, you've got to make sure your meringue is perfect. Because if you haven't got a good meringue, you're gonna struggle.
Don't flatten it. Okay, so the next ingredient that we have is oil and milk. So rather than the conventional, we use butter. For the Genoise one, we use this. Because the oil and milk, when it's emulsified together, it will actually create a very nice moist to the cake, as well as when it's combined together, react with the flour and the egg structure.
And we're adding that as our sort of like our fat component, because if we just did it as a whisk sponge without that, which we could still do, but like a Swiss roll, it has quite an eggy taste to it. It hasn't got that sort of richness to it. So we're adding the oil and the milk there for a bit of richness, that bit of fat in there. But the important thing is we're going to add that last, aren't we, to our bake?
Yes.
Because we're making this wonderful meringue. If we were to add it too early, then we might end up collapsing all of that hard work that we've done. So shall we put some things in a bowl? What have we got to do?
So what's a cake without sugar? So the first step is we're going to have the white eggs.
Do you want to use
and
this?
then we're going to whisk it with the white eggs and the meringue.
Do you need this?
Yes, put it in a stand mixer. Oh, make sure your stand mixer is clean. Maybe wipe it with a little bit of vinegar or lemon. As Josh mentioned before, if you have fat in your bake, the meringue will not rise.
I've got a couple of slides to go on that in a bit more detail as well in a minute.
So why don't, while I'm doing this, you explain about the meringue?
I've got a few slides actually. So we're going to make the meringue first. So we've got 5 egg whites in there. We've got I think do we have about 200 grams of sugar in total, but it's split.
Yes.
We've got 2/3 going in for the meringue and 1/3 which are going to go in with our egg yolks. So while Syabira gets the meringue going, which is going to make a very loud noise, I'm going to go and deliver a couple of slides now on the science behind our meringue. Then when we come back, we're going to do, it'll be bake-off style, and we'll do a proper assembly of all the bake. So I shall go and deliver a few slides now on the science behind the whisked egg whites. So I shall see you in a moment.
Enjoy the session.
So what we're going to do now, I'm going to talk a bit more detail now behind the science behind getting the perfect meringue. So what we've got here is a slide that is just showing a zoomed in look at our egg white proteins. So proteins in the raw egg white, we've got around about 20% protein in water is what we've got for our egg whites. And these are our proteins here. They're all tightly wrapped up together. I'll show the structure of that in a slide or two's time. What we're doing when we're whisking is these proteins, what we do is we denature them and we unravel them. And what you could actually do, you could actually think of proteins, think of them like a tightly wrapped ball of wool, and you've got a kitten or your cat's just come along and they've come and knocked it about and they've unraveled it all and they've all opened it up. And that's what we're thinking about here with the proteins with regards to making our meringue. So we've whisked them around, and by doing so we've unraveled our proteins, they've opened up. And then we're ending up and we're introducing these air bubbles. So you can see on this slide just here, we've got proteins, these proteins now forming around. We've got some egg white and some air bubbles in there as well. And then I'll show the next slide now, and we're going a little bit more detail. We've got more whisking, we've got more air bubbles being incorporated. And what happens is now is these proteins that have been unraveled, denatured, they begin to form around these air bubbles and they form a matrix. And it's this matrix which is making this wonderful white foam that is our stable meringue. We're also going to add some sugar into that, which you saw Syabira just add just a moment ago. That sugar is going to dissolve into the water in our egg white, and that sugar is going to help increase the viscosity. So it's going to help add a bit more body, a bit more structure, a bit more support for our meringue. So if we go a little bit further now, so that's sort of an overview of what's happening. If we go a little bit further, I'm now going to delve into what is the structure of a protein. And this is really relevant because in a little bit later in the talk, Syabira is going to be going through how proteins, they're encoded for their synthesis by DNA. So DNA, its main role, it's really important, it contains those sequences of our nitrogenous bases, and they get read and they get transferred, they get read and then they get coded to make proteins. So we're going to go through that in a few slides' time. But looking at an egg white protein, if we zoom into it, what we've got here, we've got a long sequence of amino acids. This is a peptide sequence, but they're made up of these amino acid units, these building blocks of proteins, these small sections, and each of these amino acids has got a side chain on it. And these side chains, I've just for simplicity labeled these A, B, C, and D, and you can see that I've put some as blue and some as red, and they're indicating the types of side chain, because we can get different types. So there's 20 main amino acids that make up all the proteins in our body. Some of these ones that we naturally synthesize ourselves in our body. Some of them are essential amino acids, and those ones that we get that we can't make ourselves, so we must get them from the food that we eat. But of all these amino acids, they've got different side chains. And some of these ones can be hydrophobic, some of them can be hydrophilic, some of them can be polar. And in particular, just focusing on hydrophobic and hydrophilic, you can see here, the hydrophobic ones, they're quite greasy. So they're greasy, they don't like water. We've got hydrophilic ones. These ones are polar side chains, and they like water, so they like to go towards it. And if I now show this, what it looks like for a protein in its native state, if you look on the left-hand side here, so we're looking back to those first 3 set of images that I showed you. We're zooming in on what a protein in its natural state is, and what we've got is you can see that those hydrophobic, those greasy amino acid side chains, they're all pointing to the interior of our protein. I pointed to the interior, so they're all mixed together. They all like each other because they're all nice and greasy. And that's also where, so they're all nice and greasy. And we've got all of our hydrophilic ones. They're all now pointing out towards the water because they like water and they'll interact with the water. So that's how we've got our nice stable protein. What we're doing when we're whisking now is we've unraveled this protein, and you can see now we've got this nice long chain, and we've got all of those greasy and non-greasy side chains are now exposed. But they want to now go towards what they like. So the hydrophilic ones want to go to water. The hydrophobic ones, they want to go to grease or away, as far away from the water as possible. And what they do is they look for these interfaces between what we've got is our water and our air. So you can see on the right-hand side now, when we've got these air bubbles that we've incorporated into our meringue, you can see how these proteins line up. You can see we've got all of the hydrophilic side chains of our amino acids. They're all pointing towards the water. The hydrophobic ones are pointing towards the air bubbles. And together that's making this amazing matrix, which is our meringue. That's how we're stabilizing that matrix, by putting in, by denaturing them, and they're forming in that way. So that's how our egg white comes together. I'm hoping now, if I go back to Syabira, she is going to have a perfect meringue, bake-off style, a perfect meringue that is going to perfectly demonstrate what I've just talked about.
Hand whisk the meringue, you see? No.
So there we go. So I can see that looks very nice and voluminous. So we've got a wonderful meringue.
This is what is a soft peak meringue, I would call it, because this is what we wanted for our meringue. And while Josh talking about the meringue, we actually put it in a high maximum volume on a stand mixer, put a little bit of sugar after it bubbles, and just keep on whisking it. We incorporate air into it. So now the meringue is ready. The first thing we're going to do is mix the eggs and the sugar.
So we've got 5 egg yolks there. So that's the 5 egg yolks on the 5 egg whites that we had for the meringue.
And then we mix some sugar. I like to cream the egg yolk with the sugar because sometimes when you make a lot of, in a cake where there's a lot of eggs in it, you kind of get a little bit of aftertaste of the egg yolk. But when you cream it a little bit with the sugar, it's reduced the taste of the egg yolk itself. So you can see here, just a quick whisk. You don't have to be...
Want a bit of that as well?
Oh yeah. Vanilla always must.
For flavour, bake-off, you'll have seen these make an appearance all the time. A bit of vanilla bean paste always takes things to another level, doesn't it? Vanilla goes with pretty much everything, so it's always a good addition.
So yeah, just a little bit of creaming the egg yolk. Apologies for the sound. And what we want to do next is just fold this meringue into the egg yolk. So what you want to do now here is you don't want it to be flattened. So Josh mentioned before, when we introduce a fat, protein or fat inside the meringue, it will deflate. And if you actually whisk too much when you're mixing it with the egg yolk, it will deflate. So you want to keep the consistency or the aeration of the meringue in here. Yeah?
Yep, and that's why it's quite nice to separate the eggs. So you could just, for like a normal whisk sponge, you could just put all the eggs and sugar into a bowl together and just whisk it up. That still works absolutely fine. But to get that really great volume and lightness to the sponge, it's really nice for some of these cakes where you actually separate the white from the yolk, because the yolk's got the fat in there. And if we've just got the protein, the egg white proteins on their own, then we can really get this lovely, really sort of, it's not light and fluffy. And you can see how light and fluffy that batter looks. It's wonderful.
It's very light.
Don't we just squeeze a bit of vanilla in?
Yes, go for it. So at this step, you just use your whisking and then just incorporate them all together while talking about a lot of topic. So remember, Josh, when we were on Bake Off, every single step that we do, there's a cameraman always asking us, what are you doing now?
Yeah, that's my job today. What's going on right now?
So right now, I am incorporating the egg yolk with the meringue. I'm quite nervous with the cameraman asking a lot of questions, don't you?
Yeah, well the thing is they always come round to you when something's going wrong. That's the problem. It's usually when it's going wrong, they want to go, oh, what went wrong there then, Josh? Please tell us. And then you're trying to hide what you've just messed up so they don't see it. But then unfortunately they've got their camera and they're going to go and have a little dip in there and have a little look. So that looks brilliant. We've got our flour now. Is that been sieved or do we need to sieve it? Do you want me to get you a sieve?
It is already pre-sieved flour, but I mean, for precaution, we can always sieve them.
Well, some recipes will actually say they often do a double sieving, don't they?
They do.
Because you want to make sure that all those the flour particles that we've got in there, they're all nicely separated. If you've got any clumps already in your mixture, in your flour, then they're just going to stay as clumps in your sponge batter, and then you'll have that horrible look, because when you turn your cake out and you peel off your baking parchment, and you'll just see those pockets of flour, and they really do ruin the texture of a cake. There's nothing worse than when you have that, because what will happen is the moisture of your sponge will just form like a seal around the outside of that sort of clump of flour, but there'll still be the flour just on its own in the middle, won't there?
You're going to get a surprise flour bubble when you bite into the cake.
Yes, which you don't want.
You don't want that. It's Geminoa sponge. It has to be very soft, very moist. So what you need to do when you fold your flour is scoop, slash, and scoop, and slash.
Very good.
Let me show that. Scoop, slash, and scoop, and slash. What you don't want to do is go mad like this. It will deflate the...
And we'll undo all of the hard work that we've just done.
Yes.
So did you find that as being a scientist helped you on the bake-off?
I do feel it, as a scientist, when you go into the lab, you have experiments.
Yeah.
A mission, bake a cake as an example. And then you have steps to follow. With these very little microliters, or we're working with microliters rather than a meal or 2 meals. So we're very used to follow instructions and follow the steps to execute as it is.
Yeah, definitely. I mean, I could definitely say for me, I think it was, it's that being prepared. I mean, being a chemist myself, I'll spend my day-to-day in the research lab putting on reactions, and every time I'm going to put on a reaction, I'm always going to be prepared for what I'm going to go and do. I'd never go into the lab not being prepared. I'd have written out what my procedure, my protocol is going to be. I know if I'm going to be handling lots of if there's any nasty chemicals that I'm handling, knowing all the precautions to take for all of that. And so I'm going to always go into the lab being prepared, knowing exactly what I'm going to do. So that's what I felt I took onto the bake-off. Because you've got challenges which you sort of know what you're going to make, you've got to do, you've got your 4-hour window, you just got to execute that bake in that time. So I think that's what I quite relished, really. I quite liked that, because I'm used to that bit of pressure. I'm used to sort of like going in and being really prepared for something. Yeah,
in terms of the preparation itself, this is very, very similar that we could relate to baking. It's about butter, has to be at room temperature. So in any experiments that we do, some of our setup has to be exactly at 37 degrees, some of the setup has to be 40 degrees. So we're more precise in terms of the temperature, and we know how crucial it is. And sometimes, without being in the lab, I'll probably make the mistake, don't really care much about the temperature of the butter because it is butter. We're going to incorporate it. But actually, if you try to cream a butter that in a very cold temperature, what are you going to get? Yeah,
and there's lots of other bakes where being so accurate with things like temperature is so key. Chocolate tempering being a key one, where you have got to hit very, very precise temperatures. If you go a little bit too over, a little bit under, you don't get that perfect temp on your chocolate. So I think that's where we're used to sort of working really accurately and hitting those particular points. So I think that's where it helped us quite well, doesn't it? So that looks like it's coming together really nicely now. I can't see any, there's no lumps of flour in there, so you've done very good.
No lump of flour. And then, so this is the mixture of the milk and the oil. So what I did is I just put it in the microwave for 20 seconds, high power, just to make it warm into the mixture.
And that's going to help with the emulsification, yeah?
Yes, it does help with the emulsification, and then it's rising up a little bit of the temperature of the batter.
Yep, and like we said, we're adding this last now because we've captured. You can see how light this batter is that we've made. We don't want to undo all that hard work by putting it in too early. So it's the last thing we're going to do. Our oven's preheated. We've got our tins here as well. You can see the tins, and these tins, we've lined the outside as well. For a really light sponge, delicate sponge like this, it's really great to line the outside of your sponge of your tins, because it just helps helps the sponge sort of like cling on as it tries to rise up as it bakes. So they do help really, really well. And also it just helps so you get a nice light delicate sponge at the end and get a perfect sort of lovely uniform edge around it.
Yeah.
So we've nearly got that to do. I think we've got a couple more slides for you to go into now as well, isn't it? Because we're talking, so we've folded in flour. So the flour's got flour proteins in there. We talked about egg white proteins, and I briefly mentioned about how DNA and proteins are related. So we've got that our DNA is encoding for protein synthesis. And I know we've got a few slides here for you to go through now, which is going to actually link through to how proteins are made, but also how that sort of relates to your research, because you actually did your PhD here at Leicester as well, didn't you? In
Yes.
genetics.
Yes.
So this is the amazing thing. So Syabira, she finished her PhD in 2019. I was still doing my PhD in 2019. I was in my first or second year. So actually, we were probably within 100 meters of each other for a year or two without knowing each other. This is long before Bake Off had happened. So I can pour those into the tin if you want, and then you can go through your slides if you want to.
Okay, yes. Let's switch on to the slide where I'm going to talk a little bit about how from the DNA into a protein. So in a real life scenario, when we were going into the lab, I'm working mostly into the plant genetics at the beginning of my PhD. So what important is to understand what happens in our DNA. On the slide here, you can see the first part, that the crucial part is the DNA template itself, and then after that, it will go to the transcription process, which is it involves the messenger RNA. So messenger RNA is completely copied what it is from the gene template, from the DNA template itself, and then after that, it will translate it into the amino acid sequences. Okay, to make it a little bit more clearer, and then to protein. To make it a little bit more close to the heart for our webinar today, imagine that the DNA is a recipe book. So in this recipe book today, we want to find a recipe to make Victoria sponge cake. And in this recipe book, it got a lot of different recipes. It got instruction and everything. So you can see I'll put some example there. To make a sponge, you need flour, egg, sugar, milk, and oil. To make a jam, to make a whipping cream. So all of this information that you can imagine is inside our DNA template. And then the next one is when the DNA forming from this double helix forming into a single strand, this is what we call it as a RNA transcription. For this RNA transcription, it copies everything exactly from the DNA. And then from this RNA transcription, we will go into the ribosome. So the ribosome is translating the RNA into protein process. We call it as the baker in this kitchen. So this baker actually compiling all of the ingredients that they need. They read the RNA in a codon manners. So when the ribosome trying to translate from RNA to the protein, it reads it. So A, T, G, C, and all of so on, it will have 3 amino acids together as one codon. As an example here, you can see that the ribosome C, the AUG in the human genome, is a signal to start a recipe or a signal to start protein translation. And then it will follow all of these recipes. So he knows, okay, to make a jam, I will take fruits, I will take sugar, I'll take everything I need. And then they will do the translation until it finds the stop codons. So this stop codon, it could be UAA, UAG, and UGA. So you will find all of this codon information very closely when you do your biology lesson or any kind of translation process from RNA to protein. And finally, after all of this hard work, gathering ingredients, following the recipe, once it's reached to the stop codon, there you go. That's the protein. And this protein is the worker or the final product to execute what has been instructed by the DNA itself. So in a day-to-day, I would say that this GATC is the code for everything. So when I was doing my plant science research here in Leicester, I would call it food security size matters. So what I do is I pick up the whole transcription factors family. It's a gene that we have in the plants. And then I work with these plant model species for the wheat, and we call it Brachypodium distachyon. And then we use this GATC, we use these sequences from protein to actually discover what happens during the evolution of this gene family, and then we could find out 1 or 2 candidates that affect the grain size for wheat. So this is one example how we use the genetic code as well as the protein to do the evolution study as well as unravel the characterization of any gene family. And then the second part is right now I'm doing more into bioinformatics, which is the big data analysis, Josh. Yeah. So my day-to-day life is looking at the data, either from epigenetics, which is the genome level, either mRNA, either protein. So all of these layers of information, we call it nowadays as multiomics. So people normally looking at these layers of information to understand and to characterize or to to actually look further into what could happen and what's really happening in a human genome and how these different level of protein translation actually interact with each other, either in a healthy state or either in a disease state and so on. So yeah, that's overall how I feel this GATC is a very powerful tool.
Amazing. Thank you so much for that. Right. So I have, hopefully, I've passed the test. I've evenly separated the batter into these tins. These are now going to go into the oven, and then whilst they bake, so they're going to go in they'll go for around about 30 to 40 minutes on quite a low temperature. They'll rise up really quite high as well on the tin. And whilst we do that, we're then going to move on to the decoration. So we're going to whip up some cream. We are going to make some jam. So Syabira's going to mash up some strawberries and make jam. At the same time, I'm going to go and mash up some strawberries, but I'm going to go and extract the DNA from them so they can actually see that we've been talking about DNA, but let's actually see some real DNA. Before I do that, I will also just say, if anybody's got any questions that you'd like to ask Syabira, when we get towards the end of this webinar, I'll feed them to us. So if you've got any questions, put them into the comment box, and then we'll do those at the end. Also, just to preempt you, there will be a link to a questionnaire going into the box as well at some point, and it'd be great if you could fill that in as well. So whilst they go into the oven, and you get prepped for the decoration, I shall now go and do some slides on DNA extraction from a strawberry. Thank you very much. And I shall now move station back to elsewhere in the room. Right, so if you come back to me first, what we're going to do is, hello, I'm in a different station now. We're now going to do some DNA extraction from our strawberry. So I've got the process on a slide, so if we just whiz back to the slide, you'll see what we're going to do. We've got our strawberry. You can see from this slide just here where the DNA actually is. The DNA is wrapped around, these histone protein, which make up our chromosomes. The chromosomes are in the nucleus of our plant cells, and you can see we know how small those cells actually are. So what we need to do is we need to actually do quite a bit of work to be able to pull that DNA out from there. So if we come back to me, what we're going to do is we're going to chop these up. We're going to mash them, we're going to add a bit of a lysis buffer to them, and then when we've got that, we're going to extract our DNA out. So I shall, first of all, actually, maybe before starting that, I'll say why we've used the strawberry. So the strawberry is a really great candidate for this because it is octoploid, which means it's packed full of DNA. So if you look at this slide very quickly, what we've got on this slide is this just shows it in a bit more detail. So us as humans, we're diploid. We've got two copies of each type of chromosome in each of our cells. There's actually a bumblebee, they are haploid. A banana is triploid, meaning they've got 3 sets. But the strawberry, as you can see at the bottom there, is octoploid. It is jam-packed full of DNA, and that's because that over years there was 4 key species of strawberries that have been hybridized and crossbred over many, many years, and as a result of that, they've now become octoploid. So they are jam-packed full of DNA, and they're a great candidate. So what we're going to do is we're going to do this DNA extraction process just here. So as you can see, I'll leave this slide up and we'll keep coming back and forth. So we're going to hold the strawberries. We're then going to put these into a bag. We're going to mash them up. We're going to add our extraction buffer. We're then going to mix those together, filter them off, and then we're going to add some cold ethanol, which is going to precipitate out our DNA. So if we come back to me now, let's get this started. Hopefully you can see everything that we've got. I'll move this out to the side for one moment, move these out the way. So you can see here what we've got. Here's the strawberries. I'm just going to do, I think I'll just do 2 strawberries actually. So you want to just take the tops off. We don't want those in there. So we'll just take the tops off, like so. Got 2 of those. I've got a bag here. So now we can just pop these into this bag and we can begin to mash these up. And this is an experiment that we could all do at home. So you can all do this at home tonight. You can buy a box of strawberries, you can mash them up and extract some DNA, and the rest of them you can make into jam and you can put on a scone. So you want to mash these up. I'll just take a bit of air out of that. You don't want to do it, don't be too vigorous, but you do want to mash them up into small pieces. So we'll just do that. So mash these up here, as you can see. See that just with your hands is absolutely fine. So we'll just mash that up. And that is going to help to burst open, to loosen all the cells, burst open the cells, but we're going to do that even more so with the next buffer that we're going to add. So they're all mashed up. At the same time, we're going to make our extraction buffer. And this is made now using, so we've got here, I've just got some water. There's 30 mils of water in here. I'll sit it in here just to help us show just to do this. So we're going to add some soap to this. We're going to add 1 teaspoon of soap. And I've got the structure of soap here as well to show you in a moment as well. So we're going to add 1 teaspoon of soap into here, like so. Spoon that in, like that. And then we're also going to add in 1 teaspoon of salt as well. So I've got the salt here as well. I'll just grab these. I'm going to do this as 2 half teaspoons because I've messed up the teaspoon with a soap in there. So I'm going to put, there's 1, and then there is a second one. Put those into there. That's perfect. That's finished with those. Put the lid back on this. So this is going to be our extraction buffer, our lysis buffer. And we're going to now add, we're going to just shake this up very, very slowly. You don't want to be too quick. We don't want lots of bubbles. We're going to just mix this up, and then we're going to put this into this bag of strawberries. And what's going to happen is the soap, so the soap, if I show you the structure of it, I've got this here. So this is basically a soap. This is soap like this. It's a long chain of carbon and hydrogen alternating. It's a long fatty chain, and it's got a carboxylic acid at the end. This is this C-door bond OO. So what it is, it's an amphiphilic molecule. So we've got grease this end, we've got polar this end. And what this can do is this can go into, so we've got our cell membrane. It can help, and it can find its way in. It's going to dissolve and break apart that phospholipid membrane that we've got in our cells, our cell membrane, and it's going to split it apart. It's going to burst open the cells. And once they're burst open, so it'll burst open the cell membrane, the nuclear membrane where we've got our nucleus, and that's going to release the DNA. What we've also added, we've added salt. So this is the structure here of sodium chloride, which is our table salt. This is the ionic structure of it. So we've got our salt that we've also added into here. And what that's going to do, that's just going to help so that any proteins that I've got hanging around, it's going to help them to separate from our DNA so that they're not all clumped together. So we've made this up. We're now going to add this into this bag, hopefully not making too much of a mess. So I'm going to pour that all into here. And then we're going to continue to mash this up like this. And actually, whilst I'm doing this, I can see that Syabira is getting ready making the jam. So we could probably just switch over to Syabira and we can see the jam making in action. So she's mashing up strawberries to go and make it into a jam, whilst I'm mashing it up in a different way.
I'm mashing the strawberry mercilessly so that it's all gonna dissolve and make a very nice jam. But do you know what happened to the DNA when you turn it into a jam?
What does happen? Can you tell us?
I don't know. Shall we reveal the answer a little bit later? So think about it. If the DNA being exposed to a very, very high temperature, it will disrupt the hydrogen bonds. And as a result, the DNA will be degraded. So do you think you will eat DNA while you're having the jam?
It's strange because we don't really think about when we're eating, fruits and veg and anything. You don't think about you're eating DNA, do you? When you've got your fresh strawberry, but obviously you are doing that. So it's a strange thing to think about. So I've mixed this up now, so you can see we've got this bag of mixture. What I'm going to do now is we want to sieve this. So I think for the purpose of this demo, the best way to do this is I've just got a little bowl here. I've got a sieve. I'm going to hopefully just pull this out and it doesn't make too much of a mess. So I'll just pull this out here. I've already got one in true Blue Peter style. I've got one I've already made earlier. So if it doesn't work, we'll be okay. So we'll let this just come through a little bit. I've got a spoon, I think, as well, just to help move a bit around. So we don't want all of the big lumps to go through. In fact, if we were doing something like this in a chemistry lab, if I was working in a biology lab and doing some assays, this type of experiment like this, we would actually use something called a centrifuge, which is where we can spin samples around at a really, really high speed, and it helps to separate out the solids and the liquids. So we would spin this round, and the solids would all clump at the bottom of our tube, and we would just be able to just decant off our liquid. But I don't have the luxury of a centrifuge in here with us right now. So what we've got is just a sieve. So we're going to sieve this off like that, move this over here now, and now we can pour this into a tube. So if we can get a close-up now on this wrap, we'll be able to see this. So I'll put that in there. You can see this here is our, I'll move that there a little bit. Can we see it like that? That's our soap that we used, and this here is our salt that we had. So I'm now going to just pour this here that we've got, this mixture. I'm going to pour this now straight into there. So hopefully that's around about halfway up, like that. And now we're going to add some cold ethanol. The cold ethanol, we want it nice and cold. That's going to help to precipitate out our DNA now. So I'm actually just going to put on some gloves for this because we are using an organic solvent now. So I shall just quickly pop on some gloves, and we will, and I've got some cold ethanol here. And hopefully we can, well, it's not quite as cold now because it's been out a little while. But what we can do now is you just want to really carefully just pour this just down the side of our tube, like this. Just pour it down. You want around about equal quantity of our strawberry mixture to our ethanol. And then what will happen is our DNA will begin to precipitate out. So whilst that one does that, I'll show you, this is the one exactly the same that I've just done around about 10 minutes before we started the webinar. But as you can see here, can you see that we've got these threads now of the DNA, which is actually now precipitated out. So you can see that there. That's all come out of that mixture. So it's pretty cool that we can actually see it. We've shown the structure of it on a molecular level, but now we can actually see DNA in real life now, right in front of our eyes. So that's that one there. And you can see as well with this one here, what we've just done, you can see it beginning to form and come together. Look, you can see these long white strands of the DNA that's begun to form. So if you look, so we can probably just very quickly just whiz back to the slides that we had to showcase that, and we can just have a quick summary of what we've done. So you saw that we'd mixed the contents of our extraction buffer. We poured them out through poured the slurry out. We sieved it off. Equal volumes of cold alcohol to our strawberry mixture, and we can just observe between those two lines now, those two phases, because our ethanol is less dense, so it sits on top of our other mixture, and you've seen those cloud of DNA now come together. So if we come back to me now, you can see that that's what we've got. We've got our DNA right there. So that's pretty cool. Like I say, strawberries is a great one to do this with, but you can also do things like banana, kiwi, or other really great examples of fruits that we can do it with. So that's the chemistry demonstration now. So we've seen the extraction of DNA. We've got our structures to help explain that. And now I think in perfect timing, we have now got, Syabira has got everything ready for us to do the final assembly now of our bake. So if we come back over here.
Yes, now we have our jam.
So you've done the jam very quickly. This one, we have cooled this one down a little bit now, haven't we?
Yes, we did. And then the Genoa sponge here. When you take out the Genoa sponge from the oven, thank you, Josh. So when you take out the Genoa sponge from the oven, it is important to let it cool down for like 2 to 3 minutes. And then what you do is you take out the cake and you flip it upside down. So then this is the top of the cake before, and then this is how flat it looks after you put it upside down, like this. So you're going to let it cool down, and now it's ready to be cut.
Excellent. I'll let you do that.
You won't let me do that. So this cake is actually yield about 2 6-inch Victoria sponge cake. I am going to cut it by.
Oh, we're going to go for a 6-layer cake. This is going to be exciting.
It's quite ambitious, isn't it?
Yeah, let's do it, because if we do if we do it, then it falls over. That'd be funny. But no, I think it'll look really impressive, won't it? Have you got the cream ready to whip up?
Oh no, please, Josh. It's in the fridge back there. So, 2 first layer. Oh, we're going to attempt 6 layer of 6-inch cake.
Is 1 pot enough? I can whip this up if you want, whilst you do everything else.
Okay.
Yeah. Oh, I've got some here.
So we're going to cut a cake. You can eyeball them. It doesn't have to be perfectly. Ideally, it would be nice to have the same height for everything, but if you can't, it's not the end of the world. So this is the last one. I like to cut my cake on the turntable because I can just keep my knife at the right angle and then just let it cut the cake through without have to rotate the cake. There you go. Now we got 6 layers of cake. So Josh, what is your best memory on Bake Off?
Best memory on Bake Off? Well, see, so you did this DNA Helix as your semi-final. My semi-final bake, we did a layered puff pastry dessert, and it was, I'm going to get it wrong, it's an Italian one, it was called milfogli or something like that. It's the Italian version of the milfogli. So it's big sheets of puff pastry, and then you layer that up with your sort of dip-back creams or set creams with some jams or some curds, and then you put it all together. And it was, I really quite like my patisserie, and I really enjoyed putting that one together. And it was one of those ones, I'm sure you can attest to this as well, but when you go deeper into the competition, it's really hard to practice beforehand, and so therefore I never really had enough time to nail it perfectly in the week building up to it, but on the day in the semi-final, it was absolutely perfect. And I saw Prue giving me a little clap, and she had a little jump on the spot when she walked in. So I knew I'd done an all right job with that one.
That is actually a very exciting time, isn't it?
It was really good.
I mean, being on the Bake Off is very challenging and it's very stressful, but it's only stressful up until when they say get, set, bake. But after that, you're going to be in your own zone, and then you start baking. You forget about everything. And I normally chat to myself, so the cameraman was just there to observing you, and you got a story producer asking you a lot of questions.
Exactly that.
Yeah. So I'm waiting for Josh to do his cream at the moment.
What we'll do is I'll let you finish it off because I don't want to over whip it. I'll let you do that. I've actually got a slide on whipped cream, which I will talk about very quickly. So if we quickly go back to the slides now. So this is actually what's happening when we are doing our whipped cream. So we're whipping it up, and what I love to talk about whenever I'm talking about whipped cream is I get to talk about some physical chemistry. It's actually something that will be taught if you did a chemistry degree. You cover it in your first or second year, talking about colloidal systems. And the colloid is where you've got microscopic particles suspended within another medium. So you could have tiny droplets of one liquid dispersed in another one. You could have tiny bubbles of gas dispersed in a liquid. And we've actually got both of those examples when we're whipping up our cream. So we're starting off with our cream. We have got fat globules, tiny fat globules, which are they're nicely emulsified, and they're suspended within our water. And that's shown on the left-hand side of that screen. As we then whisk it up, what we're doing, very much like the meringue, we're incorporating these air bubbles now into our whipped cream. And what's happening is we're ending up now with this really nice stiff cream. And that's now an example of we've transformed from an emulsion that we had before to start off with, and we've now formed this foam. So we've got bubbles of air suspended within this whipped cream that we've got. So it's suspended in a liquid. So it's a really nice sort of a little bit of science that's going on within our bowl. So you're nearly done with that now. I've just seen we've got a couple of questions that have actually come through. So one of them is, who inspired you to go into science, or what was maybe one of your inspirations? Could have been a teacher or somebody growing up that was your inspiration to pursue science as a career sort of ambition.
Who's inspired me during this career of science? It's a lot of inspiration. I'm taking it for all of the people around me. It is from a professor that I've learned from the university. I have one lecturer or professor that she's teaching the biology. It's like the biology living inside, like we are living in a biological world. The way that she explained how cells erupt, way that she explained how the RNA translates, it makes you more curious about what's happening in the system itself. So I think it's a lot of inspiration around me, a lot of lecture, and then it's just how it all comes together. So I would like to mention one thing, that it is an International Women's Day. And then it's important to tell all the girls out there that your curiosity is the best asset that you could get. So with this curiosity is where you would keep on asking questions, you will research more, and then when you got one answer, you're going to search more and more. So like I mentioned about Rosalind Franklin, her discovery of this DNA comes easy. Nothing comes easy, but resilience, determination, and curiosity, if you combine it together, you are unstoppable. Yeah,
definitely. And I think we've already had the video at the start, but I can say for me, Maggie was one of my biggest idols for many, many years. So for her to now be part of the university that I actually work at is even more amazing. And I've actually done live demos in this space directly to Maggie, which has been incredibly special. So she's a huge inspiration of mine. I also had a really fantastic, quite quirky chemistry teacher when I was at GCSE and A-level. He also loved his rugby as well, and I love my rugby. So he was also, I had a great conversation with him. And I think when you see someone that's teaching you a subject, and you can just tell they have that passion for it, it just helps rub off on you. And I know that definitely, there's a lot of science in my family as well, but that chemistry teacher, Mr. Reed, he was fantastic, and he definitely helped inspire me.
It's true, because I think what makes me into curiosity about this science and everything comes from your teacher, how passionate they are. And the teachers out there, thank you so much for making us today.
Yeah, definitely.
Yeah.
Right, so that looks absolutely wonderful. We can just tip that now. Can we zoom in? Oh, it's a bit bright, isn't it? We can't see. Maybe you lift a little bit up. You can see we've got.
So when you lift it up, it's like this. You don't want it to be very you don't want it over whipped. It's just enough to make into very
Yep.
smooth,
And
soft
you like to
cake.
use a mixture of whipping cream and double cream, don't you?
Yeah, I like it because sometimes the double cream can be very too thick.
Yeah, it whips up a bit too. It's because it's got such a high fat percentage
content,
in there. It can actually whip up very quickly, and you end up with it being a bit too hard to work. Whipping cream it's the perfect 1-2 whip, but sometimes it needs quite a bit of effort into it, and especially if you've got quite a humid kitchen where you're whipping it up, sometimes it can be a bit of a struggle. So actually, I think I personally haven't ever used a blender before, but having seen you do it, that's what I'm taking away from today. I'm going to start using a one-to-one mixture. And you also said that you like to just pop your tub of cream in the freezer for 15-20 minutes before you whip it up.
Yeah,
Okay.
it's whip up easier actually because you know when it's cold, so that's why the whipping cream always have to be cold because of the fat globule is easier to stabilize when it's really cold. And having it with a little bit of boost by having the frozen particle or crystal in there, it does help for them to whip up quicker. But it doesn't matter.
Excellent.
It just depends on what you prefer. But I do like put my cream in the freezer before, about 30 minutes before that.
Brilliant. Right, now we're on to the assembly. I'm going to have a very quick look to see if we've got any more questions, and I shall be back in one short moment.
Okay, so this is the attempt of 6-layer Victoria sponge cake. Dun, dun, dun. So what I do first is, as usual, you put some border around the cake to make sure that all the jam are not leaking on the side. And then what you need to do next is scoop in the jam. And then just like that. So what I normally do next is just put a dollop of cream and then just lather it on top. You call it lather, isn't it?
Yes, definitely. Yep. I'm back. I've seen we
Yeah.
had quite a few questions come through. One of them's been asking what did you enjoy most on the Bake Off itself? What was your favourite experience? That could be baking, or people that you got to meet, or along the way, challenges that ways in which you sort of like you did things on the show and that you never thought would be possible to be able to do yourself, and so pleased with it. So, what was your favourite sort of experience?
What a favourite sort of experience that I have on Bake Off? First of all, it's the people that you met. We love all the bakers. So if you watch on my year, we have a lot of funny characters like Janus, James, even Sandro. We got Carol. So it's all the people. We stay in touch with each other. And Abdul right now is away for his wedding.
Oh, is he?
Yes. And then...
Hope you've not missed it.
No, we attend his first wedding. So we did his wedding in the Northern Ireland. So we all get there, dance a nice Irish dance at night time until the middle of the night. That was much fun.
Wonderful.
So first the people that you met along the way. And second thing that I enjoy is how brave you think you would be on the show. Because on the first couple of weeks, I got a very hard week during the show, but it's only by resilience and determination and believe in yourself. So that's the second part I like about Bake Off. Once you believe in yourself and you push yourself to do whatever you believe you would like to do, somehow it happens.
Definitely.
And the satisfaction of it is unbelievable. Having your cake being criticized, not enough flavor, too dry and everything, it's just a part of it.
Yeah,
but it's all improvement, isn't it?
Yeah,
What
it was.
do you think about it?
I look back on it and think I'm not quite sure how I managed to do it, because that sort of exposure, being on a television show, the pressure of baking in that tent, it was. But also for me, because it was always a dream to go on the show, to be able to actually go there and actually do that was. It was just so surreal, walk into that tent for the first time. But I think my favourite bit that I take away from it was just, being able to construct some of those bakes, because there's so many techniques that you had to do on there, which I'd never done before. I remember on one of the weeks I had to do jelly art. I'd never ever done it before, but it gave you that opportunity to do something that you haven't done before, but you're forced to almost to experiment, to try something new. And that one, I chose the jelly art because it involved injecting different coloured jellies into that artwork.
That is actually really cool.
And actually, as a chemist, I spend my entire day putting on reactions and injecting, all of the reagents and solvents into the flask. So I thought, well, I'll do that. That's a technique that I can do. So I feel like maybe that's going to be suited to me. So actually, but then getting to do it, it was just fantastic. So I really loved doing that.
Yeah, it's just the element of experimental and so on that happens on the Bake Off itself. But one thing that I get out of from the show, it's not only on the show, one thing that I get out from the show is you realize you're getting more confident and
Oh yeah, definitely.
you know when you have a very tough meeting at work and
Yep.
sometimes you have a feedback that sometimes is quite tough, it does makes you feel like I have that recent tough feedbacks
Yeah, no,
recently.
definitely.
And I think if I wasn't on Bake Off, the resilience probably a little bit lower from what I would
That's
expect.
one of the things I often talk about. So like I mentioned at the start, I asked you about how did science or your career in science help you on the Bake Off? But I think, just like you're saying, on the reverse of that, there's so many things that I'm sure you'll say the same that you've taken from Bake Off, and it actually has now helped me in my work life, because it is being at that, challenging yourself like that and working under that much of pressure, taking on all that constructive criticism and
yes.
feedback, being able to talk and do multiple things all at once. You can take so many skills away from that. I've come back into working into the chemistry lab, doing a lot more speaking to bigger audiences now, and I've got a confidence that I never had beforehand, and that's all come from Bake Off. So it's amazing how it has really helped. This is coming together really nicely, isn't it? It looks absolutely gorgeous. I'm just seeing we've got a few more questions coming through. One of them is about when making a vegan meringue, with, for example, if you're using like aquafaba chickpea water, is it more fragile? What sort of pros and cons with it? I mean, I can definitely say, because I've made that relatively recently, the beauty of using chickpea water is the fact that actually the proteins that are in that chickpea water, they're very, very similar in the way that they exist to the proteins that are in your egg white. So they work in the same sort of way. So they denature in a very similar way. So when you're actually doing the whipping up, they're almost perfectly aligned. So actually you can get almost the same effect. So you can still whip them up. It's a little bit more fragile a foam, I find, if you've ever done it. And they take a little bit longer to bake, but I think you can still get a pretty good meringue out of them.
You can still get a pretty good meringue.
It's got a slightly different taste, which sometimes it's quite nice to. I flavour my vegan meringue quite a lot, because if you put a little bit of coconut essence or something in there, just to sort of give a different flavour, it can just help mask that slightly
taste of the.
Yeah,
I get it.
Any other questions? What else we had? We had one that is. That's a really great question. What's your favourite tool in the kitchen and then in the lab? What's your favourite implement in the kitchen that you like to use?
Spatula.
Is it? Well, and is that the same for the lab as well?
Yes, same for the lab.
Oh.
I think it's very useful tool, which is spatula. You underestimate it. You want to weigh a chemical. You want to spoon out some agar out of it, and then if you want to cook some curry, if you want to ladle. Yeah. I think versatile.
I can probably agree with. I've got a very special spatula I do all my reactions with, so I have got that. There's a few bits of quirky glassware that I've got that I really like as well in the chemistry lab. In the kitchen, a weird one, not necessarily my favourite, but the one that gets so much use, and I said this to you yesterday, I love to use cocktail sticks. They're really great because, especially just mixing in a little bit of colour into icings that you're making up. If you've got any sort of imperfections on your bakes, you've got to just knock something off or just smarten something up or correct something, they become extremely useful. So it's always worth having cocktail sticks, I find, in your kitchen. They're really, really useful. But have I got a favourite tool at home? I don't know. I've got a spatula that I really like as well, a long-handled one.
Yeah, I got a massive spatula just to fold cake.
Yep. I've got one that used to be my nan's. It's got a wooden handle and it's got a slight bend in it, but it works so well. And it's just got a lovely feel to it. It's a really nice and flexible one, so it's a big favourite of mine. What else have we got? And another question is, were we both into baking before becoming scientists? Has baking been something you've always been passionate for? Is it something that came about relatively recently?
Yeah, that's a very good question actually. This is how I get into baking. PhD is a very stressful journey, and then I get into baking when all my experiments in the lab failed for 6 months.
Really?
Yes.
And then
I have no hope. You feel like, oh, what should I do? I need to try something new.
Really?
And then end up searching for a recipe as usual. As a researcher, find all of different recipes, find out which one worked best, which one that could work in your. So I did try. So my first bake that I ever made is red velvet cake.
Really?
Yes. And that's happened to be the first technical challenge.
So yours was sort of like a bit of like an escape after having bad results in the lab. That's really strange because I've come from a completely different way. I've always loved baking. It's been a big passion for me. I've always loved it, and I see the baking and the chemistry as like one and the same. So
Ah.
I take off my lab coat at the end of the day, I go home and put the apron on, and I'm still experimenting because I love that sort of challenge and creating new things. But that's because I'm an organic chemist, and so I work in a research lab putting on reactions all the time in a fume hood. So I'm always doing, it's very similar to the setup for baking. So it's mixing little things together, heating them up into a flask, purifying them at the end. So I think there's so many parallels between the two there, which is why I think I love them so much. But no, that's fantastic. And we've got one more just come through that says,
Okay.
does having a science background help to produce a more precise and stable bake? Well, I'd definitely say from that, there's certain bakes whereby you have to be so precise.
Yes.
So, and it's where you can always say, like, it's really important for certain bake, you must follow the order of the recipe that it says. You can't just do step 1, 2, go to 5, and then come back to 3, because if you miss certain orders of additions, it can have a really big effect on the overall bake. And it's exactly the same in the chemistry lab. I can't just say, well, I'll add these 2 things first. I'll heat it up now, and I'll add this thing later on. You've got to all add things all at once. And I think that's definitely one thing that I can attest to with that.
Yeah, I do agree.
And then and regarding precision, things like, if you're doing something, I always say, like making choux pastry is one of those ones, like I always weigh out my egg for making choux pastry. I don't just say I'm using 2 eggs, I will weigh it out because I know my ratios that get me the perfect result. So I think that's where that sort of precision and accuracy comes into it.
Yeah, I do agree. And also, one thing that people normally overlook is making custard. Like, you have to temper the milk, and then you have to stir it constantly. So in a research lab, that's what we normally do. We just make sure that everything is in the right temperature, mix it slowly, and then just wait. Yeah, exactly. Patience as well.
Excellent. Well,
is a key for success.
Look at this looking stunning on there. Look at that. Wow.
There you go. A 6-layer Victoria sandwich cake.
Right, that is absolutely amazing. That would be one of those ones where you could just imagine Paul Hollywood coming through and just going right down the middle and it just flopping over, couldn't you? You're going to need a very big plate for us to eat that on. So I think that pretty much brings us to an end. And hopefully everyone's taken so much from this. We have done, we've gone through the DNA bake that we've got. We've gone through the whole journey of Bake Off. We've made a wonderful cake. We've got our, we did the DNA extraction of the strawberry. And yeah, it's been, hopefully you enjoyed it. It's been a pleasure to have you. Like I said, we have got a questionnaire that will be going, if it's not already, it will be going into the chat box at the bottom. So if you could please fill that in, that would be really fantastic, because the feedback that we get from the questionnaires is going to help us to be able to put on more webinars like this in future. So if you like what you saw and you want to see more, please do fill them in. And then we can use that, and that'll be really, really great. Yeah, and shout out to all of the girls out there. It is International Women's Day. I hope you all enjoy your life. Make sure that you utilize everything to your full potential. No one can ever beat you. Yeah, and believe in yourself. Yeah, a perfect way to end. Thank you very much. I hope you enjoyed. See you later. Bye. Bye.